Okay, now let's start to the first session for Dr. Mortuza. Dr. Mortuza, can you hear me? And are you
Yes.
ready?
Yes.
Okay. Okay, thank you very much. Before we proceed, I'd like to read the resume of Dr. Firoz. Okay, please, Mr. Rifki. Okay, this is the resume of Dr. Firoz Mortuza. Now he worked in Gamma Source Division, Institute of Food and Radiation Biology, Atomic Energy Research Establishment in Bangladesh. This is the educational qualification. He achieved his Doctor of Philosophy last year in 2020 in Tokyo University of Agriculture and Technology. He got the master degree in biochemistry and molecular biology in 2008 in University of Dhaka, and get the bachelor degree also in the same field in biochemistry and molecular biology in 2007 from the same University of Dhaka, Bangladesh. And also next, Sir Rifki. He got an award. The last award is sorry, the previous slide. He got award of the Nuclear Researcher Exchange Program at 2011 fellowship by the Ministry of Education, Culture, Sport, Science and Technology, Japan. And he worked as a scientific officer in Gamma Source Division, IFRB, AERE, Bangladesh Atomic Energy Commission from 2009 to 2014, and the position of senior scientific officer at the same organization from 2014 to 2020. And then currently he is a principal scientific officer from the same organization, Gamma Source Division, IFRB, AERE, Bangladesh Atomic Energy Commission from 2020 until now. And he has some skills like molecular biology, microbiology, and others, especially nuclear safety and radiation biology. Okay, next is, of course, there are a lot of publication that has been published by Dr. Firoz. Maybe I cannot read one by one because we have a limited time here, and maybe that's all the resume of Dr. Firoz Mortuza. Okay, Dr. Mortuza or Dr. Firoz, it's time for you to deliver your lecture. It's about 40 minutes, so the time is yours.
Assalamu alaikum. So can you hear me?
Yes, clearly.
Hi, good morning. Welcome to my presentation. My presentation title is A Biological Effect of Radiation and Application of Cobalt-60 Gamma Irradiator. I am Dr. M.D. Firoz Mortuza, working as a Principal Scientific Officer at Gamma Source Division, Institute of Food and Radiation Biology, Bangladesh Atomic Energy Commission. The contents of my presentation, first of all, I will talk about objectives, then basic information of radiation and mode of action of radiation and biological effects, then application of gamma irradiator, and activities of gamma source division in the division which I work. Objectives. First of all, I would like to talk about information about radiation, and then to improve understanding of radiation, what it is and how it interacts. The objective is to share and discuss about effects of ionizing radiation, especially gamma radiation, and to inform about the applications of cobalt-60 gamma irradiator in IFRB. Now, first, basic information of radiation. So, as we all know, radiation is a form of energy, and there are different types of radiation. Radiation is two types based on the ability to cause ionization. One is ionizing and another is non-ionizing. So suppose ultraviolet, visible, infrared, microwaves, these are non-ionizing radiation. And, and ionizing radiation is X-ray and gamma rays. Radiation can also be categorized as particle type and wave type. Particle types are alpha particle, beta particle, neutron beams, and proton beams. And electromagnetic wave type is X-ray and gamma ray. And gamma ray is emitted from the nucleus, whereas X-ray is generated outside the nucleus from the electron shell. Both of them are photons and work similarly. Now, some properties of different ionizing radiation. Here alpha radiation, it has charge +2 and a range in the air is 5 to 7 centimeter. Its health risk is ingestion and inhalation. Because it has high charge, positive charge, it cannot travel much, but it creates a very high activity or action in its small area. Then beta radiation, it has a charge of -1 and it can travel about 4 meters per MeV. And health risk is superficial skin injury. Gamma ray, it doesn't have any charge and it can travel up to 500 meters. And the problem occurs when whole body injury at high radiation, high dose. And high dose is characterized as dose more than 100 millisievert. I'll talk about this later. Now, as I said, that alpha particle, if we consider our body, it will interact in the skin and it will not pass on the skin. But beta ray, it will cross the skin and go little bit inside and it will interact there. And gamma ray can pass skin tissue, bone, and organs and can interact inside the organs. So to avoid this radiation, there are many kinds of shielding material that we can use. Suppose for alpha ray, paper can stop alpha rays, so it's no big deal. But as I said, that it is a high linear energy transferring radiation. So in a very small area, it will produce lots of ionization before it neutralizes. Then beta ray can be stopped by thin aluminum sheet, and gamma ray and X-ray can be stopped by thick lead shield, and neutrons by water or concrete. Now, units of radiation measurement. Physical units, radioactivity is measured in becquerel in SI unit. 1 becquerel is 1 disintegration per second. So if one atom breaks down or decays, then it in 1 second, then we call 1 becquerel. And from the radioactivity, we get absorbed dose. Absorbed dose is measured in gray. 1 gray is 1 joule per kg. This is a very important unit. We should remember 1 gray equal 1 joule per kg. And equivalent dose is measured in sievert, and effective dose is also measured in sievert. Now, if we think about source of radiation and the person is getting a dose from there, so absorbed dose is the amount of energy absorbed by a substance unit, a mass that received radiation. It is designated in gray. So absorbed energy divided by mass of part of receiving radiation. Now, we can measure this by a different type of dosimeter system. There are many different dosimeter system by which we can determine the amount of dose received. So from gray, then uh, we can go to equivalent dose. Equivalent dose is the expression for radiation dose in terms of which depends on types of radiation. And afterwards, because there are sensitivity different in sensitivity among organs, so we need effective dose. Effective dose is unit for expressing radiation doses in terms of effect on the human body. As I said, equivalent dose, it differs in different radiation sources. So suppose if gamma ray, X-ray, and beta particles, the radiation weighing factor is 1. So for proton beams, it's 2. For alpha particle, it's 20. So suppose if one person got absorbed dose of 1 gray, but if the radioactive source is alpha particle, then the equivalent dose will be 20 sievert. But if it is gamma ray, then the equivalent dose will be 1. Similarly, because different tissues have different sensitivity, so there is tissue weighing factor, and red bone marrow, colon, lungs, stomach, breast. These are highly sensitive, so the tissue weighing factor is 0.12, highest for these organs. So if we multiply the tissue weighing factor with the equivalent dose, then we will get the effective dose. And by the summation of all organ effective dose, we can get the effective dose of the whole body. Now, where is radiation? It is not that radiation is only in the nuclear facilities like this. Radiation is inside a human body. The most important natural radionuclides that are found in human body are uranium, thorium, polonium, lead, potassium, carbon, beryllium, etc. So we have these radionuclides inside our body. Now, if we only consider potassium, a normal person contains about 140 grams of potassium, among which the radioactive potassium, the abundance is 1.2 into 10 to the power minus 4. So if we calculate, then we can get 4,375 becquerel from only potassium, radioactive potassium. So radiation dose per year from potassium, one person, normal person, he will, with a body weight of 70 kg, will get 1.3 into 10 to the power minus 4 gray radiation from potassium only. And other radionuclides are also there. Now, inside, as I said, that there is radiation inside our body. In the environment, we are getting radiation from many sources. Suppose cosmogenic radionuclides, we get 0.1 millisievert dose from there. And from our body also, we get about 0.26 or 4 millisievert. And from the terrestrial radiation, like thorium from uranium and other sources, we get about 0.3 millisievert. And we inhale radon. Radon comes from uranium and other sources. So we get radiation of 1 millisievert from radon per year. and cosmic radiation is about 0.28 millisievert. So in the environment, there are many sources of radiation that we are exposed to. A human being absorbs about 3 millisievert dose per year from nature. Now, how does it act? The mode of action of radiation and biological effect. Now, I will talk about on the gamma ray and X-rays. X-ray and gamma ray, first of all, they are high energy photons, so they will dislodge electron from atom and make it ionize, and this is the direct effect. And afterwards, the electron with the energy of the photon, it will create ionization, reacting with other molecules, especially water, because in the cell about 70% is water. So it will mainly react with water, and it will produce different type of free radicals and ions like hydroxyl ion, hydrogen ion, and hydrogen free radical, hydroxyl free radical. So these free radicals and ions, it will interact. It will interact with the biomolecules, other biomolecules. Now, why? Because short life of these free radicals and high activity of these free radicals, and they only those formed in water column of 2 to 3 nanometer around DNA are able to participate in indirect effect. Now, because they are, as I said, that they are very reactive and they have short life. So in 3 nanometer proximity, if the DNA is close to the ion that are formed, free radicals that are formed, then it will cause or it will react with the DNA. One thing is that why we are concerned only about DNA mainly. The reason is DNA is the most important biomolecule of our cell. Suppose if proteins or carbohydrates or lipids are damaged, then from DNA and other sources, they can be reproduced. But if DNA is damaged, then there is only one copy, so it cannot be reproduced. So we are mainly concerned about DNA. But the free radical or hydroxyl ion that are formed, it will affect other biomolecules as well. It will affect not only in DNA, it will affect with protein and carbohydrate and lipids and others also. Now, types of DNA lesion that are formed. There are different types of DNA breakdown or damage that occurs, but most lethal is the double-stranded DNA break, where two strands are broken, and single-strand DNA breaks are also dangerous. Now, suppose a person, if a cell or a person is got a 1 gray dose of gamma radiation, then how much damage it will cause. Now, 1 gray of dose will cause base damage of 1,000 to 2,000, and deoxyribose damage about 1,200, and single-stranded breaks will be there about 1,000. Double-stranded break, which is most important and most critical, it will produce 20 to 40 actually double-strand breaks in the DNA if someone is exposed to 1 gray of radiation. But this 1 gray radiation is very high actually. There is no possibility of getting that much radiation except a nuclear accidents. Now, what happened in the cell? A cell has complex signal transduction and cell cycle checkpoints and repair pathways to respond to DNA damage. So when there is damage, then our cell has the DNA repair mechanism. So it will repair the DNA. And if it is incomplete repair, then mutation occurs. And then possibility of causing cancer and hereditary effect. And if repair fails, if there is lots of breakdown and a repair enzyme cannot repair the DNA, then cell death occurs, and which can lead to possibility of causing acute effect and fatal effect. And if repair succeeds, then there is no hazard. In human body, there is antioxidant process to neutralize free radical. The free radical that is produced by radiation and other sources. So we have an antioxidant process which neutralizes them. And also there is very efficient DNA repair system to repair DNA damage. Now this work was done by some researcher, and it is a very, very good article. So the researcher showed that suppose these are human cancerous cell line. So these are pre-irradiation cells. Before irradiation, these are the picture of before irradiation, and these are the dose 100 milligray, 500 milligray, and 1000 milligray. These are suppose marker which binds to those positions where there is double-stranded break, BSB. Like here, yellow fluorescent protein is tagged with tumor suppressor protein, PP3BP1. So these proteins bind to the double-strand breaks. So by this way, these are called foci. So by the number of foci, we can understand how many double-strand breaks are there. To say it in simply, that by the number of foci, we can say how many double-strand breaks are there. So as we can see that suppose if there is 100 milligray dose, then and there are about two double-strand breaks, and it increases with increasing dose. So in 1000 milligray or 1 gray, 1000 milligray means 1 gray, there will be about 20 to more than 20 double-stranded breaks. So, however, the researcher showed that, as you can see in this picture, that these are the doses, and over time, suppose at 4 hours, as you can see that the repair mechanism, it repaired many. Suppose there was two double-strand break and it repaired. There were many double-strand breaks here, but it repaired some of them. And by this way, with the graph, we can see that with time, the number of double-strand breaks is repaired. And the researcher showed that suppose 24 hours or after 48 hours, the cell recovers and repairs all the double-stranded breaks. Now, we have things to compare, that what causes cancer. We have some understanding that radiation causes mutation and it causes cancer. Yeah, it's true. It's true to some extent, and it is dependent on the amount of radiation. But there are also other things that we should know. That, as I said, as I showed in previous slides, that 1 gray of radiation, it produces base damage of 1000 to 2000, and single-stranded breaks about 1000, and double-stranded breaks 20 to 40, which is very lethal. Now, in our body, there is cellular metabolism, and to produce energy and to do other things, cell do metabolism. So during this normal phenomena of cellular metabolism, every day in each cell there is about more than 12,000 base damage occurs, and more than 55,000 single-stranded breaks occurs, and more than about 25 double-stranded breaks occur every day per cell per day. And this was mentioned in the article in Cell, published in Cell. So So 2 to 3% of all metabolized oxygen is converted to free radical during cellular metabolism. And about 70,000 lesion is produced per cell per day due to cellular metabolism. This results in an average of about 1 mutation per cell per day, whereas background radiation of 1 millisievert per year produces uh, about 10 to the power minus 7 mutation per cell per day. So DNA damage, DNA breakdown, mutation, these things are all natural and it occurs inside our body. Now, I will talk about application of gamma irradiator. Now, gamma irradiator are equipped with high activity radioactive sources, mainly cobalt-60 and cesium-137, which are used for different purposes such as sterilization or decontamination, etc. Now, there are many applications of gamma irradiator. It can be used for sterilization of medical product, pharmaceutical product, and tissue-based biological product, and also in radiotherapy and cosmetics and toiletries. It is used in research and development, and also mainly also in food irradiation. Now I will focus on food irradiation here a little bit. Food irradiation is a technology which applies ionizing radiation uh, to food for improving the safety of food and extending the shelf life of food by reducing or eliminating microorganisms and insects. Now, food irradiation is a very safe process. There are also many other processes for decontamination, like heat or chemical, but researchers found and the policy-making bodies, they found that food irradiation is very safe, and it is permitted in over 60 countries, and about 500,000 metric tons of food are processed annually worldwide. As I said, a very safe process. I said it because because by food irradiation, when we irradiate a food, uh, it makes some chemical changes or like this, but it does not make the food radioactive. It doesn't make the food radioactive. This is one important thing. It is like that when we irradiate something, it's like we switch on the light so we can see everything. But when we switch off, there is no light comes from the things that we could see. So what I'm trying to say that by irradiation, the food or anything, it will not become radioactive. And from there will be no radiation. There will be no radiation. So it is very safe. Now, in the world, there are many sources that are used in food irradiation. Gamma rays are used, X-ray and electron beams are used also for food irradiation. Now, irradiation can serve many purposes. In case of food, now prevention of foodborne illness. To effectively eliminate organisms that cause foodborne illness, we can use food irradiation. We can use it for preservation also, to destroy or inactivate organisms that cause spoilage and decomposition and extend the shelf life of food. It can also use for control of insect to destroy insect, and it can also be used for delay of sprouting and ripening, to inhibit sprouting in potatoes and delay ripening of fruit to increase longevity. And it can also be used for sterilization of food for the use of patient like AIDS patient, and also the NASA use this kind of food for the astronauts. Now, same thing, almost same thing here. The dose is mentioned. Now, for low dose irradiation up to 1 kilogram, it is called radicidation. And the purpose of this is to inhibit sprouting, delay in ripening, prevent insect infestation, and parasite control and inactivation. So they use different doses. All of them are below or equal to 1 kilogram. And there is medium dose, 1 to 10 kilogram. And this process is called radiation. And purpose is extend shelf life of raw and fresh fish, like etc. Extend shelf life of refrigerated and frozen meat products, and reduce risk of pathogenic and spoilage microbes in meat, seafood, spices, and poultry, etc. And increase juice yield and reduction in cooking time of dried vegetables, etc. So for this purpose, uh, radiation use of dose from 1 to 10, and for high dose above 10 kilogram, it is called rad appartization. And mainly it is used for sterilization of spices, dry vegetable seasoning, sterilization of packaging material, sterilization of food uh, uh, who are immunosuppressant patient. So for those people, food are irradiated and sterilized and then given to them. So for this purpose, a high dose is needed. So the dose is above 10 kilogram. Now I will talk about my organization, my institute. So I work in Gamma Source Division and Institute of Food and Radiation Biology. We have cobalt 60 gamma irradiator. Cobalt 60 is produced from cobalt 59 by heating cobalt 59 with a neutron. It produces cobalt 60. So this cobalt 60 decays to nickel, and in this decay process, it releases one beta of 0.31 mega electron volt and two gamma electrons, two gamma rays with high energy, 1.17 mega electron volt and 1.33 mega electron volt. These energies of gamma is used for different purposes like sterilization and decontamination in our division. Now, initially, a cobalt 60 gamma source was brought from Canada in 1980. The strength of the source was 50 kci, and that is 1,850 terabecquerel during commencement. Afterwards, it was successfully disposed to the Health Physics and Waste Management Unit at our organization. This is the first irradiator, and now it is decomposed in our Health Physics and Waste Management Unit. Later in year 2000, 50 kci cobalt 60 batch type gamma irradiator was installed by BAEC India. This is the irradiator. Actually, this irradiator is mainly research oriented, and we give semi-commercial service also. So it is not very high. The activity is not very high, only 50 kci in the year of 2000. Afterwards, in 2014, the activity was increased to 90 kci, and that is 3,330 terabecquerel in cooperation with India. At present, the source activity is 35.4 kci. Now, we put our products like this, and we irradiate it for certain times, and afterwards we do dosimetry. How much radiation it got. So for dose measurement system, there are 4 dose measurement system that we use in our division. A Frique dosimeter, it is for the low dose dosimetry, 5 to 400 gray. It is in gray level. It determines dose of gray level. Others like amber perspex, red perspex, steric Ceres dosimeter. These dosimeters are used for high dose radiation, like above 1 to 30 or 50 kilogram dose. Determine how much dose the product received. We use this kind of dosimetry system. These are some of the picture of dosimeter. So now the irradiation services of GSD, activities of Gamma Source Division. We We give a gamma irradiation service basically for in two sectors, in research and development purpose. We give it at free of cost, and for commercial purpose to earn revenue. And the stakeholders for research and development are Institute of BAEC, Bangladesh Atomic Energy Commission, where I work, and other research organizations all over the country comes to our division for research irradiation purpose. And we, as I said, that we also use this source for semi-commercial purpose. And many food industry and medical and pharmaceutical companies come to our facility to take irradiation service. R&D services. Gamma Source Division provide irradiation service for research to different private and government organizations, including Institute of BAEC for free of cost. List of irradiated research sample of different Institute of BAEC. These are different institute and divisions of Bangladesh Atomic Energy Commission. They use different samples for R&D purposes like fruit fly, pupae, cucumber, silkworm eggs, microbial solution, male banana bud, and pupae and blowfly pupae, dried fish, fresh fish, bitter gourd, pineapple. This is my food technology division. And the tissue banking and biomaterial research, they irradiate amnion graft, bone graft, bacterial suspension, tissue graft, etc. Other institutes also use their samples for irradiation. And there are different doses for this research. And these are the list of irradiated research sample of different organizations of Bangladesh. These are apart from BAEC, other organizations like Bangladesh Forest Research Institute, many universities, Bangladesh Sugarcane Research Institute, Bangladesh Institute of Nuclear Agriculture, Machine, and many other institutes send their product to our division, and we irradiate it according to their demand. And as you see, there are different types of product: wood block, fish, sugarcane seed, rice seed, peanut, etc. that we irradiate. Commercial service, we give commercial services to different companies. They send their spices, raw materials, surgical materials, and injection and syringe, and different types of pharmaceutical products. And also there are many different types of food products like spirulina, turmeric, mushroom, and bull stick for pet food, and goat ear for pet food. These kind of products they send for irradiation. So Gamma Source Division provide irradiation service to commercial products of different medical and pharmaceutical companies and food processing and manufacturing companies of Bangladesh, and earns revenue from for Bangladesh. In last 5 years, Gamma Source Division earned about 20 million taka from this. And these actually the main purpose is to provide service to the companies. So there are many pharmaceutical and food processing companies that send their different types of materials in our division for different purposes. And these are some of the list of the companies and their products. And this is also other companies, more than 50 pharmaceutical and food processing company, they come to our division for radiation purpose. Summary, Gamma Source Division is providing irradiation service to R&D samples at free of cost, and GEG earns revenue from the country by rendering irradiation service to commercial product. And the demand and scope are increasing significantly day by day. And so the number of irradiators and necessary facilities are going to be increased in near future in Bangladesh. Now, as I discussed earlier about radiation, uh, so there are three basic rules when there is any radioactive source or when we hear about radiation, we should try to follow this rule. This is uh, time, distance, and shielding. We should uh, spend very less time, uh, as less as possible, um, and we should try to maintain distance from the source, and we should try to use proper shielding for to protect us from radiation. Now, the issue of radiation exposure is not only a matter of safety, it is also a matter of benefit compared with risk. Because yes, there is little bit risk, obviously, but we should also think about the benefits of radiation. As, but and we need to be cautious and careful about radiation, but we should not be frightened unnecessarily. This is one important thing that we should remember, that we have to be very careful and cautious, but we should not be frightened unnecessarily. Now, for the students, I would like to have some question like this. I hope that they will be able to answer this question. Which organ is most sensitive to radiation? And what is the main source of mutation or cancer? Radiation or cellular metabolism? And does food become radioactive after gamma irradiation? And is it safe to eat gamma irradiated food? These are very simple questions. Thank you very much. In Bangla, we say apnake onek dhonnobad, and in Indonesia, maybe it's terima kasih banyak. Thank you very much for your time.
Okay, thank you very much, Dr. Firoz. It's very interesting presentation, and we got a lot of new information, especially about the irradiation process, how we calculate, and we know that from your lecture, we know that the gamma ray has more penetration than the alpha and beta ray, and also radiation is everywhere, right? Even in human body, and it is radiation process is natural occur in human body, and for the food, radiation to food doesn't make the food become radioactive. That's I think important statement. So we don't have to scare to consume the radiated food. Okay, now I will open the discussion session. So if you have any question, you can raise your hand or write your question in the chat box.
Dr. Firoz, I want to talk some question.
Yeah.
Okay.
Thank you very much, Dr. Firoz Mortuza, for your nice presentation. Can you please show me the slide, double stand break, single stand break, base damage? You give the rate money.
Yeah. This one or this one?
This one, yes, this one. So here, please show the please.
Yeah.
Okay.
Can you see?
Yeah. Here you showed that base damage, single stand break, double stand break. This slide is for natural radiation or radiation from external source.
Yeah, of course it is from external source, because it is not possible to get 1 gray radiation from natural source.
Nature.
If someone is exposed to that much radiation, suppose if nuclear disaster occurs,
Okay.
or if was for experiment, research related experiment, if we expose a cell of a human cell to 1 gray of radiation, then this type of DNA damage occurs.
This radiation, if somebody somebody is exposed 1 grade radiation at instantly, may you mean that?
Yes.
We know that one cancer patient every day he's exposed on an average 2 gray.
Yes.
So if anybody exposed more than 1 grade, that is 2 gray, possibility of occurring single stent, double stent, or base damage is more than this. Okay.
Of course.
Oh, okay.
Okay.
Cellular base damage. What does it mean?
Yes, there are 4 bases of DNA, like adenine, guanine, cytosine, and thymine. So, it will, and there are 4 nucleotides, and there are purine and pyrimidine bases. So it will damage those bases, it is the double strand.
Yeah.
Now here these are the bases.
Oh, okay. Thank you.
Okay. Thank you very much, Mr. Fakharuddin, and thank you for the answer from Dr. Firoz. Okay, anyone has other question? You can raise your hand and I will select.
I want to ask.
Okay.
Okay. So high energy
I'm
gamma
sorry.
radiation produces electron disruption.
I'm sorry. Please, can you introduce yourself first and then the question?
Okay. I apologize. My name is Adela Devra Fransna. I'm from TIP. I'm from new university student.
Okay, please continue your question.
Okay, so like Sir said, high energy gamma radiation produces electron disruption that can cause this disruption result in damage to the DNA and other cellular structures. So can it degrade such products like food with high fat content? And can it I think that's my question.
Can you please repeat again and a little bit slowly, please, and clearly?
Okay. Can you repeat maybe slowly, because the maybe I can
Okay. Convey.
So the high energy gamma radiation produce electron disruption or ionization.
Yes.
That cause this disruption result in damage to DNA, right?
Yes.
And other cellular structure. So can it degrade such product like food with the high fat content?
Actually, the purpose of food irradiation, suppose decontamination or as I said, that sprout inhibition or like this, the amount of gamma radiation that we use, it will not change the nutritional value very much, because very little amount of protein or carbohydrate or lipids and this thing will be changed due to this much radiation. But microorganisms like suppose bacteria will be killed, because if we can compare the size of a protein and a carbohydrate in comparison with the size of bacteria, uh, uh, so uh, the bacteria is huge. So the gamma radiation, the amount of gamma radiation that will kill the bacteria, but that much gamma radiation will cause a very little amount of proteins or carbohydrates or lipids to modify or change. There will be change, obviously. There will be damage, but the amount is very, very little.
Okay, so basically, food irradiation is safe in a certain dose, right? So
Of course, because whatever we do, suppose we will not eat something raw. Um, so we cook, we use many things to avoid contamination. So even if we cook, then there will be also a production of free radicals. There will be a production of ions and this thing. So in every cases, in every way, there is a possibility of denaturation or changing of proteins and carbohydrates and this thing. Um, so gamma rays also do the same thing, and actually it is more, it is safer. It is safer than suppose heating or using chemicals and this thing for decontamination and other purpose, sterilization or other purposes.
okay, so how about the answer?
I think that's enough, but may I ask other question?
Okay, maybe we will move to another uh, the other question before we can start before we continue to your second question. Okay, so we'll give
Okay.
chance to other for the
Okay,
question.
okay. Okay, Dr. Firoz Mortuza, I have another several question. Another question. Hello,
Yes.
Dr.
Okay, yes, Mr. Pakharudin.
Okay, I in 2014 uh, I attended one IA training program in Malaysia. So here from IA training program I know that radiation causes cancer. It is not established uh, till now. It is not established that radiation causes cancer. So, um, what's your observation in this comment from IIA information?
Actually, as far I know, radiation can cause, obviously, radiation can cause cancer. Definitely, but it will depend on the dose of radiation. It will depend on the dose. And actually, there are so many parameters, because as I said that in the natural metabolism process, there are many DNA damage and this occurs also naturally. And our lifestyle, our food behavior, eating behavior, and these things also have many effects in DNA and suppose mutation and these things. So it is difficult to understand exactly from where the cancer is coming from. But as a source, suppose as a source of, like we say, carcinogenic source, radiation is obviously a carcinogenic source. It can cause cancer at a certain dose, like other chemical carcinogens and this.
I want to add here that my observation is that radiation may enhance the probability of cancer. Directly radiation causes cancer. I think
till now it is not established. It may enhance the probability of cancer.
Yeah, as I said that suppose because the normal process is going on, and every day and every time the cell, whether it is getting radiation or not, there is denylation and these things. So it is difficult to separate from which source it is coming. So it is, as you said also, it is difficult to say, but radiation is a cancer causing agent, obviously. Cancer causing agent, but the amount of is matter here.
Okay. Thank you very much for the answer, Dr. Firoz. We have another question in the chat box. I will read that question. This is from Dr. Anggoro. I'm sure he is from the Gajah Mada University. Thank you for interesting sharing, Dr. Firoz. Although many experts have convinced the public that irradiated products are safe for consumption, what is the perception and acceptance of people in Bangladesh about these food products?
Actually, public perception all over the world is almost same, and especially in developing countries or underdeveloped countries, the public perception is not very good. Actually, people does not know about radiation that much, but there is the one thing is that here the food that are irradiated, mostly these irradiated products are for import purpose, so for export purpose. And in Bangladesh, normally we don't use irradiated product of our country, but the public perception is not clear actually. From Bangladesh perspective, it is not clear. However, there is no problem of acceptance till now. That there is no question arise due to food products which are irradiated in Bangladesh.
Okay, thank you for the answer. And I think this is the last question because the time is almost over. This is from Miss Ragilia. The question is, can the curd contaminated by tapeworms still be repaired? How much redurization does to overcome the problem of meat contaminated with tapeworms? Does it affect the taste change? Okay, so meat. Oh, can the meat, not curd, can the meat contaminated with tapeworms still be repaired using the radiation?
Okay. Now the thing is that meat is irradiated by gamma irradiation, gamma source. So before in Bangladesh, in my division, suppose before we irradiate something, how much dose is needed for that particular product, we do dose measurement. We give different radiation dose first time. Suppose 1 kilogram, 5 kilogram, 10 kilogram, and up to 25 and 30 kilogram. And afterwards, we check that how much radiation is needed to sterilize that product, or to get the desired result. So suppose for tapeworm, the similar type of experiment needed to be done, that how much radiation needs to be given to decontaminate tapeworms. And I think within 1 to 10 kilogray, it will die. So there will be a little bit taste change, but it will not affect that much.
Okay. Thank you very much, Dr. Firoz. Actually, we still have some question and one raised hand, but unfortunately, the time is now is 10:24. The time is for Dr. Mortuza for this first session is already over. Maybe we can continue the discussion outside this forum. Okay. Thank
you
you.
very much. Yeah.
Yes. Thank you.
I would like to especially thank Firoz and the organizers, and also the students and other participants. I have very little knowledge actually, but thank you very much for listening my lecture. I hope you all the best.
Okay, thank you very much for the lecture and explanation in discussion session. We really hope to invite you, especially in offline event in the future. Okay.